Good afternoon, everybody. Hello, everyone.
1 minute here. Yeah.
So, I hope each and every one of you is doing good in the pink of health and happiness. So, we'll begin the class today. Today, we are going to learn about cell cycle and cell division. So, we'll go through the PYQs one by one, and along with the PYQs, we'll also go through the topics of that particular PYQ, the concept of that particular PYQ. Is everything good? Can we start? Am I audible, visible? Would you give just yes? Thank you, Kiran. Thank you. So yes, we'll begin the class with the first PYQ, that is the match the list one. We need to match it, choose the correct answer from the options. So here cells are metabolically active and proliferate in or DNA
G1 phase.
yes, and DNA replication takes place in S phase.
Yes.
Protein synthesize in
G2 phase.
G2, and quiescent phase is G0.
G0.
And here in the quiescent phase, one of the most important thing you need to remember is, in the quiescent phase, the cells are metabolically active. Okay, if they make a if they make the question saying the metabolically inactive, if the yes, very good. So if it is if they ask metabolically inactive, is it true or false? In quiescent phase, the cell is metabolically inactive. Is it true or false?
False.
False. So here we make A2 B4, that is D4 Delhi as a correct answer. Here following are the stages of cell division, we need to make it into correct sequence. First is G1,
G1 phase.
yes, G2, then M phase. After M phase, that is karyokinesis, M phase is karyokinesis, and after karyokinesis it is cytokinesis. So which is the right answer?
Option D.
Okay, E C, yes, this one, B for Bombay is the correct answer. ECADB, very good. Among the eukaryotes, replication of DNA takes place in, where the replication of DNA takes place? It is very easy as that. Yes, we can move on to the next one, that is S phase, that synthesis phase, where synthesis takes place in, synthesis of DNA takes place in S phase. Now given below are two statements. Statement 1, during G0 phase of the cell cycle, the cell is metabolically inactive. They have asked metabolically inactive, is it correct? Actually it is false. Metabolically they are active. The centrosome undergoes duplication during the S phase of the interphase. Yes, during the S phase of the during the S phase of the interphase, what and all happens? DNA replication and centriole or centrosome, whatever you call it, centriole duplication also takes place during the S phase. So which makes statement 2 as correct. So what is the right answer?
Option
Statement
A.
1 is incorrect, 2 is correct. Very good. Match the list. M phase is quiescent phase, is inactive phase. Inactive in a sense here, not metabolically inactive, it is cell division wise it is inactive. That is what they meant here, inactive, quiescent phase. G1, interval between mitosis and the initiation of DNA replication, that is G1 is between the, no, not G1, sorry. G1 is between the synthesis phase and previously it is the M phase. previously M phase, and from M phase it goes to G1, from G1 it is S phase. So, it is interval between the mitosis and the initiation of DNA replication, that is G1. G2, proteins are synthesized. M phase, it is, yes, it is equational division. Very good. So, what is the answer?
Option D.
Option D. D for Delhi. Yes. Which one of the following is quiescent phase? Easy as that. Which is that?
G0.
G0 phase.
G0.
Very good, very good. G0. This is a very, very good question. The fruit fly has 8 chromosomes. That is 2n is 8 in each cell. Okay. During interphase of the mitosis, if the number of chromosome at the G1 is 8, okay, G1 is 8, what would be the number of chromosome at the S phase? S phase, what is the number of chromosome?
8.
8. Yeah, from here to here, the chromosome number is same, but the DNA content is
Double.
the DNA content is doubled. Very good, the DNA content is double. So, it is very easy, you do not have to even write anything, it is that easy. It is D for Delhi is the correct answer, 8 chromosome. Chromosome number is same in the equational division, the chromosome number is same throughout the life cycle of the cell. The centriole undergoes a duplication during
S phase.
S phase, we just discussed. Again and again they have asked us, very easy question, again and again they have asked us. It is the third time we are doing this. S phase mein DNA replication, G2 protein synthesis, quiescent inactive, G1 if you see they have not even changed this thing, isn't it? List 1 and list 2 same to same they have put cut and pasted in the next year. So, this is the one. So, it is 1 4 1 2 3.
Option B.
Option B. They have only rearranged the options. Very good. Identify the correct statement with regard to G1. G1 phase में reorganization of all the cell component takes place. Is it? Where it takes place? Reorganization of all the cell components.
G2.
No, actually reorganization of all the cell components, it is the most dramatic phase. Which is the most dramatic phase? Most dramatic phase of the cell cycle is M phase. Not metaphase, M phase, mitosis phase, that is cell division, all like PMAT, that is prophase, metaphase, anaphase, telophase, all the phase, whole total make M phase. Okay, so here is the most dramatic phase, reorganization of everything. Okay, if it is like it आधा करता है ना, if it is 20 mitochondria, 10 each. If how much content of Golgi apparatus will be there? It will be divided. All the dramatic phase will be taking place. That is reorganization of all the cell component takes place in the M phase, not the G1. Cell is metabolically active, grows but does not replicate its DNA. G1, yes. Nuclear division takes place. Where nuclear division takes place? Again in the M phase. Nuclear division matlab karyokinesis.
Correct.
Yes. And DNA synthesis or replication takes place in the S phase. So, we need to say the correct statement for G1, that is cell is metabolically active, grows but does not replicate its DNA. Is it correct? B for Bombay? Yes. Where the replication takes place? Is there any lag? Is there any lag? Subhash, is there any lag? Would you check? No, yes, yes.
Visible only. It's visible and the audio voice also check on.
Yes, yes. Thank you so much. So it is B4 Bombay is the correct answer. Some dividing cell exit the cell cycle and enter the vegetative inactive stage. This is called quiescent phase or G0. The process occurs at the end of where actually it occurs. If this is a, I'll tell you.
G1 phase,
Yes.
M phase,
No, no, no. Where is it? Okay, fine. If this is the cell cycle, if this is M phase, G1, S, G2. Okay. So, here cytokinesis takes place, in this cytokinesis takes place and it enters the G1, from the G1 it exit to G0. If it has to enter, then again it will enter to the G1. Understood? So, here where exactly it goes out of the cycle, it will be from the G1 and where it can come back to the cycle, that is also G1. Understood? Here. Any doubts, children? Any doubts? So, what is the answer now? The answer is A, that is G1 phase.
G1,
Match the following events that occurs in their respective phases of the cell cycle. Again almost same. Yes, G1 phase. S phase is a DNA replication and chromosome duplication. Metaphase in the M phase, alignment of the chromosome, and G1 cytoplasmic growth takes place. G2 cell growth and organelle duplication. So, yes, 2, 2, yes, G1. So, this is the thing.
option C,
Option C. G1 cell growth and organelle duplication, yes, cytoplasmic growth, yes, one. But the thing is, in G1 phase, cell growth and organelle duplication, almost maximum of organelles will be duplicated in the G1 phase. In G2 phase, only some like mitochondria and some other will be duplicated, and also here protein synthesis also takes place. What type of protein? The protein like tubulin. What are these tubulin proteins? Actually, these tubulin proteins are helping in in formation of spindle fibers and all. Those are the tubulin, especially such type of proteins will be synthesized here, and here also protein synthesis takes place. What kind of proteins here? Like DNA polymerase and all other proteins which will be helping enzymes, making enzymes, helping in the DNA replication will be synthesized here in the G1 phase. The correct sequence of the phase of cell cycle is easy.
option D,
Is easy as that. Option D. Cell in the G0 phases. Enter the cell cycle. No. Suspend and terminate the cell cycle. No. Cell in the G0 phase actually exit the cell cycle. DNA replication in bacteria occurs in? It's a very good question. DNA replication in bacteria occurs in which, where exactly? Tell me. Is it in the S phase? See, DNA replication in the bacteria doesn't actually this one, the phase, the cell cycle, we call it as amitosis. Amitosis are direct cell division. So there is no such thing called S phase. Okay, no such thing called G1, G2, S phase, such things. Directly from one cell, you know binary fission, right? If this is a bacterial cell, what it does? It undergo binary fission to make two cell. There is nothing such G1, G2, S phase, mitosis, nothing takes place. This is called direct or amitosis. So prior to the fission, actually DNA replication takes place like this. If this is the nucleoid, prior to the fission, this is fission. Prior to the fission, the DNA replication takes place here. In this phase, actually DNA replication takes place. So answer to this question is prior to the fission. Okay, because here they have clearly mentioned bacteria, not eukaryotic cell. Only in the eukaryotic cells, even in the eukaryotic cells like amoeba and all undergo amitosis. Amoeba, paramecium, or like protist, especially protozoans, all undergo amitosis only. The higher cells, higher organism, plant cells, animal cells, so these undergo proper mitosis. The binary fission in amoeba, you might have learnt that also undergo amitosis. Binary fission of leishmania, you might have learnt, that's also undergo amitosis. Paramecium also undergo amitosis. Such things. Understood? So this is the answer. Answer is C. During the cell growth, DNA synthesis takes place in easy
S phase.
S phase. So many times they have asked, right? In the previous year, this is a fourth question we might be answering one-liners. A somatic cell that has just completed the S phase of its cell cycle as compared to the gamete of the same species has, this is really good question. Tell me. The somatic cell has, which means it is a diploid cell. Okay. The gamete cell is a haploid cell. The somatic cell has just completed the S phase. S phase in the sense, here the content is doubled, content is twice, the number of chromosome same.
Same.
No, we have to compare for the haploid cell. This is gamete cell, and this is somatic cell. Now it is 2n is the number of chromosome and the content of DNA is 4C. If you say 2C here it is C is the content of DNA, and if it undergo mitosis, then it will be N, after the S phase, it will be, the content will be 2C. Now tell me.
Option D.
No, not same number of the chromosome, not same. See, listen. We have to compare a gamete cell to the somatic cell. Okay. Now this is the gamete cell which has content of the DNA is only C. Okay, content. And the number of chromosome is N. Here the number of chromosome is 2n, the content is 2c. Now it has undergo the synthesis phase. The content has become 4c and the number is still 2n. So compared to this, C, here it is 4c, N, here it is 2n. So the content of DNA has become 4 here, 4 times. And this one, the number of chromosome has become twice. Understood? We will do it again. The somatic cell that has just completed the S phase of its cell cycle as compared to the gamete of the same species. The gamete is always haploid in number, somatic cell is diploid in number. Now the somatic cell is undergoing the S phase. Now the somatic cell which has undergone the S phase, what is the number of chromosome? Number of chromosome is still 2n. What is the content of chromosome? It is 4c now. As compared to the gamete, Gamete has a number of chromosome N and the content is C only. Okay, we will take the number, some number I will take. Somatic cell has, for example, Mano 8 chromosome and content of DNA is any number you give, or we will take 2C only as they have given 2C. Okay, the content before the S phase. Now after the S phase, same number of chromosome will be there, that is 8, and content will be doubled, that is 4C. The gamete of the same species will be having half of the number, like 4 will be the number in the gamete, and what is the content of DNA? Content of DNA is only C. So, with respect to the 4, it is double the chromosome is doubled. With respect to C, it is 4 times, that is the content is 4 times. Understood? That twice the number of chromosome and 4 times the amount of DNA. This will be the correct answer, A. Any doubts regarding this? Tell me. This is very good question actually. Any doubts, or shall we move on? Yes. During which phase of the cell cycle amount of DNA in the cell remains at 4C level if the initial amount is denoted as 2C? Tell me. In which one? In G1 it will be 2C, in S it will become 4C, and it will become, not in, it will become 4C, especially in G2 it will be 4C. So, in which phase of cell amount of DNA in the cell remains at 4C level if the initial amount is 2C? It will be only G2. Now, it will become 4C here. So, do not take in the S also, G2 and S. Which is the answer? It will be D4 Delhi. In G2 phase, the amount of DNA will be, actually you can see it is doubled, 4C wala. In S phase of the cell cycle, amount of DNA is reduced to half in each cell. No, actually it is doubled. Amount of DNA doubles in each cell. Same nahi, chromosome number never increase. So, it is B for Bombay is the right answer. Spindle fibers attach to kinetochores of chromosome during which phase?
Metaphase.
Very good. D for Delhi at metaphase, because it will come to the equatorial plate like this. So, for that, for making it to come to the equatorial plate, it has to attach the spindle fibers, and also for the division. With respect to the metaphase, which of the following statement is incorrect? Chromosome lie at the, we need to say for the metaphase. Chromosome lie at the equator of the cell? Yes. The complete disintegration of nuclear envelope takes place? Chromosome are highly condensed? Yes. Metaphase chromosome are made up of 4 sister chromatids held together by centromere? Is it 4 sister chromatids? Actually, if this is the centromere, this is one sister chromatid, this is one sister chromatid, and this is another sister chromatid. How many sister chromatids are here? 2 sister chromatids. So, which makes D for Delhi as a wrong answer, as a wrong statement. Which one of the following never occurs during the mitotic cell division? Never occurs during the mitotic. Coiling and condensation of chromatids? Yes, it does occur. Spindle fibers attached to kinetochores of the chromosome? Yes. Movement of centrioles towards the opposite poles? Yes, it happens. Pairing of homologous chromosome? Why should it has to pair? Only in the meiosis this happens, pairing of the homologous chromosome. Like if it is chromosome 1, chromosome 1, it has a pair. So, these two pairs actually come together for crossing over, for recombination, for that. So, if this is the chromosome 1, this is a homologous chromosome, this is also chromosome 1, this is also chromosome 1. Chromosome 1 of the chromosome 1 will be coming from the maternal side, that is mother, another one will be coming from the paternal side, from here this thing. So, we all have a pair of chromosome. So, this homologous chromosome, that is same type of chromosome coming together, this will take place in the meiosis, not in the mitosis. So, D for Delhi is the correct answer. Select the incorrect statement with reference to mitosis. Incorrect. Splitting of centromere occur at the anaphase is, remember this, in mitosis, especially in the mitosis, splitting of centromere occurs at the anaphase. If this is the chromosome, how exactly the splitting takes place? How exactly the splitting takes place? Like this. Like this, the splitting takes place. Splitting, the centromere here, the centromere will be split and it will be moved to the opposite poles, with this will be moving this wise and this will be moving this side. Understood? The splitting of centromere takes place in the anaphase. Okay, there are 3 anaphases we can see, I mean in including meiosis. In that anaphase of the mitosis, splitting of centromere takes place. Anaphase 1 of the meiosis, when I am talking about the meiosis, does the splitting of centromere takes place? Yes or no, tell me?
No, ma'am.
No, splitting of the centromere will not be taking place, because this homologous chromosome, one of the homologous chromosome will be going to one end, another one will be the whole total chromosome itself will be going with the centromere intact, will be going. So, anaphase 1 mein splitting will not be taking place. Anaphase 2, will the splitting taking place?
Yes, ma'am.
Yes, it will be taking place. So, you need to remember this. Okay, there are so many previous year PYQs have been asked on particular this concept about the anaphase only. In mitosis, in anaphase, splitting of centromere takes place. In meiosis 1 mein it is not taking place, in meiosis 2 it is taking place. Remember it. So, what is the, okay. So, we need to find the incorrect statement. All the chromosome lie at the equatorial metaphase, yes. Spindle fibers attached to centromere mein attach होता है क्या?
No, ma'am.
It will be kinetochore. Very good, kinetochore. It will be attached to kinetochore, which means this is the wrong answer. Wrong statement. Chromosome decondense in telophase, it happens. So, it is C is the right answer. Again, correct sequence. This is second or third time we are doing it. Just tell me. G1 is G2 and M.
Option A.
Option A. Very good. Easy. Attachment of spindle fibers to kinetochore becomes evident in, yes, metaphase. Which of the option gives the correct sequence of the event? This is very good question. Condensation, nuclear membrane disassembly, mitosis mein crossing over doesn't happen. Condensation, nuclear membrane disassembly, arrangement at the equator, centromere division, segregation, telophase, yes, this is correct. Condensation, crossing over, no. Condensation, arrangement, centromere division, same to same, yes. Segregation, telophase. Okay, this is also true, but B becomes more right answer because the nuclear membrane disassembly is written, especially in the prophase it happens. So, which is the correct answer now? Both this B can also be correct answer, D can also be a correct answer, but which is more correct between B and D?
B.
Yes, B for Bombay is the more correct answer, because you can see nuclear membrane disassembly is also written here. Anaphase. Anaphase promoting complex. Okay, this is Ames question. Okay, anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in human cell, which of the following is expected to occur? So, in anaphase what happens actually? The segregation of chromosome takes place. Segregation. Okay. In the metaphase, this will be arranged in the equatorial plate, right? In the anaphase, this will be pulling to each other, this thing, plate. Okay. So, before that, till here in the metaphase, there will be something called securin is the protein which will be inhibiting that segregation. Okay, segregation को inhibit करके रखता है. So that is a, this will be inhibiting the segregation. So this is the inhibitor protein. APC, that is anaphase promoting complex, comes here and tells this securin, अभी तेरा काम खत्म हो गया है, अभी ज़्यादा inhibit मत करो. Actually it will be inhibiting the protein called separin, जो separate करता है, separin. Okay, it will be inhibiting this one, inhibiting कर रहे. So APC comes and tells, अभी तुम हटो यहाँ रास्ते से. Allow the separin to separate the chromosome from this centromere से centromere को अलग करके separate करने के लिए बता दो. This separin will do it. So if APC is not working properly, what happens? Will the separin do the separate? No, क्योंकि separin को inhibit कर रहा है securin. That's why the chromosome will not segregate. If you are not remembering all these things, it's okay, fine, don't remember it. Just for the explanation, I gave this explanation. So what is the answer for this? It is chromosome will not segregate. Segregate in the sense, from here वो ऐसे खींच के इधर इस तरफ नहीं जाता, यहाँ एक खींच के इस तरफ नहीं आता. समझ आ रहा है? Understanding? Actually, this APC has to tell securin to stop doing its duty, inhibition duty को खत्म करो, allow the separin to do its duty, that is separation. That has to be, that command is actually given by this principle, that is APC. If APC is not working properly, so nothing else will work properly. That is segregate doesn't happen. Understood? If you understood, just give a thumbs up. Thank you. Which of the following is not a characteristic feature during the mitosis in somatic cells? Not the characteristic feature. That is spindle fibers formation, yes, will be taking place. Disappearance of nucleolus will be taking place. Chromosome movement, yes, it will be taking place. What about the synapses? Synapses will be taking place in meiosis. So it's D for Delhi is the right answer. Understood? Spindle fibers attached to, tell me, just now, I mean you have already given the answer to this question. Kinetochore of chromosome, that is B for Bombay. Here they have given kinetosome something. Okay, please when we look at it, we tend to sometimes नज़रअंदाज कर देते ऐसे चीज़ों को, तो please be cautious about when you are taking the right answer. A cell at the telophase stage is observed by a student. Oh, it's a very good question. Yes. A cell at the telophase stage is observed by a student in a plant brought from a field. He tells his teacher that this cell is not like other cells at telophase stage. There is no formation of cell plate. Okay, plant, what happens in the telophase, especially in the plant cell, the cell plate this is one nucleus, this is another nucleus. It is not like the animal phase which furrow takes place, it is the plant cell. This is the cell plate formation. Cell plate formation takes place, and there is no formation of cell plate in this particular thing. There is no formation of cell plate containing more number of chromosome as compared to the other device. This would result in, if there is no cytokinesis, which means what they are trying to tell is no cytokinesis. Karyokinesis has happened, but no cytokinesis. So, what is the number of chromosome in this? If there is, particularly if in the 2n condition, if the plant has, for example, 8 chromosome, manke chalo 8 chromosome, now this has 8 chromosome, this has 8 chromosome. Now the chromosome are double. This particular scenario, situation, what do we call it as? Aneuploidy? No. Polyteny? No. Polyploidy. Ploid ko na multiple we are into, I mean multiplying it, that is called polyploidy. This particular situation we call it as polyploidy because there is no cytokinesis. Cytokinesis होना था, इसको अलग सेल बनाना था, लेकिन ऐसे नहीं हुआ, तो it's a condition called polyploidy. It's a very good question. Yes, this a stage in the cell division is shown in picture. Select the answer which gives correct identification of the stage with its characteristics. Which phase is this? Actually, this is yes, telophase. Now, what is its characteristics now? Endoplasmic reticulum and nucleus is not formed yet. Nahin. Telophase nuclear envelope reforms, Golgi complex reforms. Late, no, this is not late anaphase, no. So, will be the most appropriate answer will be B for Bombay is a right answer. Given below are two statements, chromosome become gradually visible under the, okay, we are going into the meiosis part. Gradually visible under the light microscope during the leptotin stage. Yes, it is correct. See, L for L in the leptotin, L aata hai na, it will be visible under the light microscope. L, ऐसे याद रखना कि in leptotin stage, yes, the cells will be visible as not cells. It is the chromosome. Chromosomes will be gradually becoming visible under the light microscope. We can see in the leptotin stage. Yes, that is correct. The beginning of the diplotin stage is recognized by dissolution of, not dissolution, actually formation. Oh, sorry, correct. I'm really sorry. Yes, the beginning of the diplotin stage is recognized by the dissolution of synaptominal complex. Yes, actually where the formation, I will tell you. This meiosis 1, in the meiosis 1, it is divided into prophase, metaphase, anaphase and telophase. So, in the prophase, the prophase is prophase 1 especially. What is it divided into? It is divided into 5 sub phases. What are those sub phases? That is, we'll remember it as a Lata Zara Pani De Do. Lata Zara Pani De Do is a mnemonic, we keep it as, and that is a sequence. L for leptotin. Okay, what exactly? Yes, this exactly takes place. Condensation starts and they can be becoming visible under the light microscope, especially in the zygotin. This two homologous chromosome will be coming together. If chromosome 1 has pair, right, another pair will be there. That pair, I mean another one will be coming close. If this is chromosome 1, this is chromosome 1, they will be coming close. Okay, when they are coming close, they'll there is a proteinaceous complex, that is synaptominal complex, they will be binded like this. Okay, so formation of synaptominal complex. Complex takes place in the zygote, synaptominal complex. So if this is, I mean, if this is one chromosome like this, this is another one chromosome, this is two, right? They are coming like this. So we can form the bivalent. This is one chromosome, this is another chromosome, that is chromosome 1 chi pair. We can see the bivalent, or else it's the synaptominal complex. After that we can see the tetrad. Okay, if this is chromosome 1 ka one chromosome, chromosome 2 ka another chromosome, there is a synaptominal complex forming the in between these two. So we can see this arm 1, 2, 3 and 4. So this makes the tetrad. Otherwise this is one chromosome, another chromosome, otherwise we call it as bivalent chromosome, bivalent. So that particular stage, bivalent and tetrad, you can see it in the zygotine stage. Now coming to the Packettine stage. Packettine, what is the major thing happening? It is crossing over. Crossing
over.
Yes, very good, crossing over. Otherwise we call it as recombination. Recombination. So this in the zygotine stage, anyway, they have come close and joined. Okay, synaptominal complex se aise join ho gaya. So now crossing over, how the crossing over takes place? From the one arm, the crossing over takes place like this. Synaptominal complex is still joined hai, but the crossing over takes place like this. Okay, that is recombination. ये हाथ यहाँ आ जाएगा, ये हाथ यहाँ आ जाएगा when I remove it. Okay, so this is the crossing over and the, but synaptominal complex is still there, na? It is attached like this. Now coming to the diplotin stage. Diplotin stage, what happens? Dissolution of synaptominal complex. Dissolution of synaptominal complex. So here I will tell you, first the formation of this one chromosome takes place in leptotin. It became visible. Then they came together to form the synaptominal complex. In Packettine, the crossing over has taken place. In the diplotin, this synaptominal complex was still there, right? Now the synaptominal complex has broken. Okay, but still this crossing over ka this point, they were still together in this point. This particular point where they are still together, this is called chiasma. This is called chiasma. In dissolution of synaptominal complex takes place, but they are still attached at chiasma. Otherwise you can call it as chiasmata. Okay, now the dissolution has happened, but they are at the point of contact still like this, This is called chiasmata or chiasma. And during the diakinesis stage, how they will be segregating like this. The chromatids will be going, but they will be still attaching at the tip. This is called terminal position. ये position terminal ना, terminal मतलब final last position, but they still be attached like this. They were like this. Then they became dissociation of synaptominal complex, but they still at the chiasma in the diplotin. Then they are moving towards the end like this, terminalization. We call it as terminalization, and they separate. So in the diakinesis, terminalization happen. Terminalization of chiasma. The chiasma is the point of contact. Now terminalization of chiasma takes place. Then they separate. So understood? Let us zara pani de do. Leptotin, zygotin, packettine, diplotin and diakinesis. If you understood, just give me the thumbs up. Rest didn't understood, only Surabhi understood. Chris, thank you Chris. Sakshi. Yes, very good. So this is about it, because this is very important. Leptotin mein condensation, and we can see it under light microscope. Zygotin mein formation of synaptominal complex, packettine mein crossing over and recombination, डिप्लोटीन में डिसोसिएशन ऑफ सिनोप्टोमिनल कॉम्प्लेक्स बट दे स्टिल अटैच्ड एट द chiasma. Diakinesis में terminalization of that chiasma is taking place. This is very important. We can see when we are going to the next statement. Okay, what is the answer to this? Both are correct. Both statement 1 are true. Okay, C is the correct answer. Match it. Diakinesis, completion of terminalization of chiasma. Yes. And pecketin, appearance of recombination nodule. Recombination takes place in pecketin. Zygotin, synoptominal complex formation takes place. Leptotin, chromosome looks like thin threads. They can be visible under the microscope. So it is
Option A.
2, 4, option A. Very good. Recombination between homologous chromosome is completed at the end. Recombination between homologous chromosome is completed by the end of recombination. Recombination where it takes place? Recombination takes place in pecketin. So after by the end of the pecketin, the recombination has already taken place. What is recombination? This is the chromosome and this is the another chromosome, homologous. When the recombination takes place in this particular part, right? So how it will become? Particular this part has become the recombination. So this will takes place in the pecketin, even end also, pecketin stage. So that is pecketin stage. Match it. Chiasmata formation takes place in the diplotin. Crossing over is pecketin. Synoptomimal complex formation will be taking place in the zygotin. Terminalization of chiasma will be the diakinesis. So the things which I wrote here, right? So that is very important and they have asked so many times. Just remember 2 to 3 points of each division of Prophase 1. That is enough to solve the question. Understanding, students? So what is the answer now? 3-1. 4-2. Yes, this is A is the correct answer. Any doubts? Just tell me any doubts. Any doubts? No doubts. Yes. Which of the following stages of meiosis involve division of centromere? Division of centromere will be in the
D option.
Diffodeli. Yes, anaphase 2. What happens in the anaphase 1? Anaphase 1 doesn't have any division of centromere. The process of appearance of recombination nodules occurs. Very easy. Recombination nodule कहाँ आता है? Recombination कहाँ होता है? Pecketin, yes. Select the correct statements. The tetrad formation is seen during leptotin. Is it? Tetrad formation is seen during zygotin. So this makes the statement as wrong. During anaphase, the centromere split and the chromatids separate. Yes. Terminalization takes place during the pecketin. Where the terminalization takes place? In the diakinesis terminalization takes place, not in the pecketin. Nucleolus, Golgi complex, ER are reformed during the telophase. Is it correct? Yes, it is correct. Crossing over takes place between sister chromatids of homologous chromosome. The crossing over takes place between the
Non-sister.
non-sister chromatids. So this is wrong, which makes B and C as a right answer. We need to find the right answer, correct answer here. So B and C. Oh, sorry, B and D. B and D as a correct answer. So which is C is the right answer. Any doubts? The dissolution of synaptominal complex occurs during, tell me where?
Diakinesis.
Diakinesis terminalization happens. Okay, terminalization hai na? So if this is crossing over, then dissolution of synaptominal complex, here dissolution takes place in the diplotin. Dissolution of synaptominal complex, diplotin, okay, not diakinesis. Diakinesis में terminalization of chiasma or chiasmata, okay, that is diakinesis. Diplotin में it will be dissolution of synaptominal complex. Understood? During which stage of the mitosis and meiosis respectively does the centromere of each chromosome split? सेंट्रोमियर कहाँ स्प्लिट होते हैं? एक तो एनाफेज में होते हैं एंड मियोसिस में एनाफेज 2 में होता है. एनाफेज 1 में तो कोई सेंट्रोमियर स्प्लिट नहीं होता. सो इट इज एनाफेज एंड एनाफेज 1. एनाफेज ऑफ द mitosis and anaphase 2 of the meiosis 2. So here the centromere splits. Select correct sequence. लता जरा पानी दे दो. DABEC.
Option.
Yes, option C. This stage of the meiosis can last for months or years in the oocytes of some vertebrates. Actually, so when we know the leptotin, zygotin, packaging, diplotin and diakinesis, in particularly in the diplotin stage, the arrest in this particular diplotin stage, there will be arrest of oocytes. Oocytes will be arrested in this stage. Okay, when the embryo will be forming, when the fetus will be forming, in most of the vertebrates, even in humans, even in human female. So in the diplotin stage, there will be arrest. In this particular stage, diplotin stage, arrest of oocytes will be taking place. Okay, so you might have learned in the reproduction in humans chapter. So at puberty, this will be going to the next stage, that is diakinesis. Then it will be entering the meiosis. Then it will be again arrested in the metaphase 2. Metaphase 2. Yes. So where exactly it will be arresting? That will be diplotin stage. Many vertebrates do that. Okay, at birth they will be having certain number of eggs which is already formed. At puberty, one after the other, the eggs will be starting maturing. Only after the fertilization, actually after it will undergo second division, that is second polar body will be released only after the fertilization. So we will be learning in human reproduction, but here this is important, that is diplotin stage. In meiosis, crossing over and exchange of material between the homologous chromosome catalyzed by the enzyme. So what is this crossing over? This is crossing over means recombination. What is the enzyme used here? That enzyme will be recombinase. easy as that, easy to, nothing to remember. Recombination will be taking place, and the enzymes which helping in recombination is recombinase. Actually it cut the part of the DNA from one non-sister chromatid, and it will cut the part of the DNA, then crossing over from here it will be joining this one to that in another one, from this one it will be joining to another one, that is non-sister chromatids mein cut and joining. It cuts and joins, which means it has two this thing. It can also cut, it can act as nuclease also, and it can act as ligase also. Ligase, it is a molecular glue, ligase, and nuclease, it can act as nuclease, that is cutting. It can also act as joining, that is ligase, both, that is recombinase. Identify the correct sequence of events during the prophase 1. Correct sequence, you just, it is jumbled. Identify the correct sequence. First it will be chromosomes gradually, yes, tell me. Leptotin, this is leptotin. Synapses of the chromosomes, synapses of the homologous chromosome is zygote. Crossing over between non-sister chromatid is a pectin. Terminalization of chiasma, diakinesis. Dissolution of synaptomal complexes,
Diplotin.
diplotin. So just arrange it, B A C E D, that is D for Delhi is the correct answer. Bivalent or tetrad formation is a characteristic feature observed during
Zygote.
yes, chiasma in the zygote, no. Synaptomal complex in the zygote, yes. Chiasma in the diplotin, no. Diplotin mein pachatin nahi. So it is synaptomal complex in the zygote. You can see that the formation of tetrad or bivalent chromosome. The appearance of recombination nodule, again, yes, it is in the, where it happens, actually it happens in the
Pectin.
pectin stage. Which is a pectin stage now? Terminalization is a diakinesis. This is the zygote. Bivalent is a zygote. Cited with a crossing over takes place is the pectin. So it is D for Delhi is the correct answer. So just remembering that 5 things about the sub classes, not this one, division, subdivision of the prophase 1. So many questions we could solve, right? So you just need to remember 5 things under the 5 subdivision. Understood? Regarding meiosis, which of the statement is incorrect? Need to find incorrect. 4 haploid cells are formed at the end of the meiosis 2. Is it correct? Yes, it is correct. There are 2 stages in the meiosis, that is 1 and 2. We know that. Yes, it is correct. DNA replication occurs in the S phase of meiosis 2. Actually, before meiosis 1, S phase happens. Pairing of the homologous chromosome and recombination occur at meiosis 1. Meiosis 1, that is pectin, that is prophase 1. Prophase 1 mein, that is in pectin, prophase 1. This happens. So this is also true. So which makes C as a wrong statement, incorrect statement, because DNA replication occurs in the S phase of the meiosis 1 और meiosis 2 से पहले कोई S phase G1 G2 कोई नहीं आता. There is a short lived interphase, we call it as very short lived, where the DNA decondensation also not properly takes place. So there is no S phase, there is no G1 G2 before the meiosis 1. What is the sequence of it? It will be G1 S G2 तुरंत it is meiosis 1. meiosis 1 के बाद ना there is some interphase interphase, it is very short, very short. Complete decondensation also doesn't take place. Okay, next happens meiosis 2, meiosis 2. Okay, so it is a C is a wrong statement. Which stage of meiotic prophase show the terminalization of chiasmata? You know that. Tell me.
Diakinesis.
Diakinesis. Very good. Which of the following stage of meiosis involve division of centromere? Again and again they have asked this question. Centromere, we need division of centromere. Anaphase 1 में होता है क्या? नहीं, it will be anaphase 2. See, it is such a easy question. They have not even given anaphase 1 also. See, the options could be the anaphase could be the answer, which is of mitosis, and anaphase 1 of the meiosis 1. So that could be the option. The option itself is not even confusing. मतलब ऐसे option ही set नहीं कर रहे. So it is so easy. It has become so easy, isn't it? Don't you think, children? मतलब अच्छा खासा options भी दे सकते थे. So they just want you to score marks. मतलब exam setters है ना, exam setters set कर रहे हैं ना paper. उनको ये मानना है कि स्टूडेंट्स शुड गेट द मार्क्स, दे वांट यू टू गेट द मार्क्स, इसीलिए ऐसे वाला ऑप्शंस रखे. ऑप्शंस इटसेल्फ इज नॉट जस्ट कंफ्यूजिंग, मतलब इतना आसानी से इट्स इजी. ओके फाइन. मैच द फॉलोइंग विथ रिस्पेक्ट टू meiosis. Again, like they don't have any other questions to ask. Like how many, this is the 4th time we are solving this question, right? Like that easy now. Zygote, synapses, easy. Packaging, crossing over. Diplotin, chiasma. Diakinesis, terminalization. Easy. What is the answer now? 4, 3, 2, 1.
A.
Yes. Very good. So now are you getting the confidence to solve the questions? Are you? I hope you might have got the confidence right now, isn't it? So whatever the question comes, no, you could definitely solve it. After we will be solving around 60 questions of previous year question, and you know how pattern, you got to know the pattern how they are setting the questions. So now you should get the confidence solving this. Dissolution of synaptomal complex occur. मतलब कोई और क्वेश्चन ही नहीं है इसमें, बताओ. Dissolution of synaptomal complex कहाँ है? पता नहीं, ये डिप्लोटिन. Okay, no questions. देखो, the first 5 things which I said: leptotin, light microscope, zygote, formation of synaptomal complex, that is tetrad and bivalent. Packaging is a crossing over recombination. Diplotin, that is dissolution of synaptomal complex as well as formation the chiasma. At the chiasma they will be joining chiasma. And diakinesis, terminalization of chiasma. सिर्फ यही 5 वर्ड्स के इर्द गिर्द ही घूम रहा है क्वेश्चन. ओके, नथिंग मच. यस. ड्यूरिंग meiosis 1, in which stage the synapsis takes place? Tell me. Synapsis takes place in the zygote, that is synaptomal complex takes place. The stage during which separation of paired homologous chromosome begins. Separation of paired homologous chromosome, that is, ऐसे थे, क्रॉसिंग ओवर हो गया, अभी सेपरेशन हो रहा है. बता दो. Paired homologous chromosome. Separation of paired. The separation starts, begins बोल रहे, begins. हाँ, you should see now this ends ना, diakinesis में terminalization होके end हो रहा है, begin कहाँ हो रहा है, begins will be in the डिप्लो. easy question है लेकिन confuse होके, मतलब question को ठीक से पढ़ो, ठीक है, because you'll be in the hurry, 3 hours का एग्ज़ाम है and you'll be so much hurry, इसीलिए आधा अधूरा क्वेश्चन मत पढ़ो, पूरा क्वेश्चन पढ़ो, क्योंकि 4 मार्क्स है यार. ये देखो का 2018 में आस्ड है. सो बिगिन्स पूछा है इधर, एंड नहीं, एंड प्रेमोनाइजेशन हैपेंस इन द डायकाइनेसिस, लेकिन बिगिन कहाँ होता है, सिनोटर्मिनल कॉम्प्लेक्स ऐसे टूटता है ना, टूटता कहाँ है, इट विल बी इन द डिप्लोटिन, नॉट द डायकाइनेसिस. डायकाइनेसिस में एंड होता है, ठीक है, ठीक से, वेरी इजी, लेकिन ठीक से क्वेश्चन पढ़ो. बता दो अगेन, दिस इज द 100th टाइम वी आर सॉल्विंग दिस क्वेश्चन. बता दो. Pectin will be crossing over. Metaphase 1, yeah, chromosome align at the equatorial plate, and equatorial plate में ना 2 ऐसे 2 equatorial plate form होता है. Metaphase 1 में 2 equatorial plate form होता है. Okay. Diakinesis, terminalization, zygotin, pairing of the homologous chromosome. Yes, it is 3, 4, 2, 1. That is C. Easy, very very easy. In meiosis crossing over is initiated at, crossing over कहाँ initiate, crossing over होता कहाँ है, initiated तो मतलब ये बहलाने के लिए, मतलब ये करने के लिए दिया है. Where exactly crossing over takes place? Crossing over takes place at the Pectin. Easy as that. Select the correct option. हाँ, synopsis align the homologous chromosomes. Where? Zygotin. Synthesis of RNA and protein. G2. Action of enzyme recombinase. Recombination. The recombination कहाँ होता है? Pectin. Centromeres do not separate but chromatids move towards the opposite pole. Centromere, where it doesn't separate? It doesn't separate in the anaphase 1. Centromeres do not separate in the anaphase 1, which makes 2, 3, 5, 4. D for Delhi as the right answer. Arrange the following events of meiosis in correct sequence. Crossing over, Pectin, synapses, zygotin, terminalization of chiasma, diakinesis, disappearance of nucleolus. It will be also after the diakinesis, yes. So this will be B, A, C, D, B, C, D. Yes?
Option A.
Very good. Yes, very good. The enzyme recombinase is required at which stage?
Pectin.
Pectin. Answer here का सवाल, ठीक है. The complex formed by the pair of synapsed homologous chromosome is called
Valent.
bivalent. Easy as that. So this is the end of PYQs. पिछले 13 years का PYQs हैं. So around 60 questions we did it. So any doubts regarding this? This is the end of the PYQs. Any doubts regarding the this one cell cycle, cell division chapter? Any particular topic you didn't understood? You can ask me. You can switch on your microphones and tell me. Otherwise, you just tell me how are you feeling about the class, whether you are getting the confidence after solving so many PYQs or not. Just tell me, because till now we didn't get enough of time for any interaction, right? So today we have got the time. Like 4:15 may we'll end the class. Like 3 minutes are there, so just share your opinion so that I'll also get to know that how are you feeling, and within like next 23 days mein you are going to write NEET exam. Anybody wants to say something? It was such an interactive class. I really liked the class. So just give me a thumbs up whether you have got the confidence regarding the chapter. Surabhi, thank you. Chesta, Kiran, thank you so much, children. That is what needed, okay, required. We need to have confidence when we are solving the question. So one of few important like suggestions which I want to give is, one thing is properly read the question, okay, properly from starting to ending, properly read the question. When they have asked incorrect, okay, if they ask incorrect statement, you underline the incorrect one. I mean, incorrect होता है ना, उसको underline पहले करो, so that we need to always keep in mind that we have to find the incorrect one, because in the options incorrect भी एक option में दे दो, like BCD is a incorrect, BCD is a incorrect, and A and E is the correct one. So they will be giving both the options. So we need to keep in our mind that what we have to take the incorrect ones. So because of that, in the question पढ़ते ही incorrect को underline कर दो, तो कि it will be in our head, in our mind, that we have to find the incorrect one. One of the first this thing advice about it. The second advice is, even if you find the correct answer, अगर 4 option मान लो कि पहला option में ही हमें correct answer मिल गया, but बाकी के 3 भी पढ़ के देख लो, क्योंकि we need to find more correct. Some of the questions you might have already saw where we are finding more correct ones. There could be 2 options which we feel correct, but we need to find more correct. अगर पहले ही तुमने पहले ही question option में correct answer मिल गया, or if you're not reading the next 4, there is chances of you losing marks. So ऐसे मतलब don't give so much of time, but just read the questions. It may take around like 10 seconds भी नहीं लेता. Other 4 options, you just look into it. Okay. Because we should be more sure when we are marking, because each and every marks counts. ठीक है, 4 मार्क्स, अगर 4 मार्क्स की जगह तुम्हें -1 हो रहा है, इट इज़ लाइक 5 मार्क्स लूज हो गए ना. 4 मार्क्स आना था, उसमें तो रॉन्ग करके -1 आ रहा है, तो वी लॉस्ट 5 मार्क्स एट होल टोटल. सो दैट इज़ नॉट वेरी गुड. स्पेशली इन बायोलॉजी यू शुड टारगेट more than 330 मार्क्स टारगेट करना ही है आउट ऑफ 360. मोर देन 330 आना ही आना है. समझ में आ रहा है? Because the competition is so much and it is easy to score. You saw the previous year question, so easy to score. We just want to be very clear about the concept and we have to read the question clearly. बच्चे यही गलती करते हैं कि they don't read the question clearly. ठंडे दिमाग से सोचना चाहिए. इतना you have to keep your head cool all the time when you are reading the question. एग्ज़ाम हॉल को पहले ही पहुँचना, आधा घंटे पहले ही वहाँ रहो. Be relaxed. When you get the question paper, be relaxed and start reading it. Understood? So that is the end of the chapter, this one, today's discussion and session. So if you liked it, just give me a thumbs up.